having low throughput when compared to like next method. Actually, in PCR based marker, you take SSR or RFLP, you need, it's a laborious. Even in SSR is less laborious than RFLP, but SSR also laborious. You know, to do one marker, for example, you take, you are doing some QTL mapping studies, QTL mapping, you're having nearly 200 population. 200 population, you are getting nearly 100 polymorphic markers. You need to run 100 polymorphic marker on the 200 and plus 2 parents. 202 means 202 PCR's reaction means in one PCR plate, how many will fit in? All three are parents plus maybe 93 individuals you may get. No, no, six parents and 90 genotypes you can at a single go can do PCR of one marker, means for every, to cover entire population you have to do two times PCR. Two plates you need to keep for one marker. Two plates you have to do PCR for one marker to cover 202 individuals. Means for example, you have to do 100 mark. Means you have to keep 200 plates. Then 200 plates means 200 gels we need to run. You know, at least even though our lab, actually when I was working in RICE, our lab is very good lab. We're having 78, I think, gel electrophoresis, big actually, can able to very big gel electrophoresis. Although at least max to max, if you're efficient, very good enough, you can run 6 gels, 6 plates. Even though you are working nearly 8 to 10 hours continuously, if nobody is working in the lab. 8 to 10. To cover 200 plate, how many days you require? And you know, every time if you're making gel, either it coming good or no, you don't know. Bands will come or I don't know. So very laborious. How much marker you are going to cover? Only 100 marker, only 200. Sometimes you may, it's not, have to run nearly 3 to 400 marker, nearly 500 to 6,000 population. Then what you will do? Like in a simple situation, had to do this much work. It is entire your PhD work. It will become. That's why nowadays many are shifting to more advanced marker. And when it is come to coverage, see max to max, how many polymorphic marker you will going to get? 10 to 12 or 13. Max to max 20 you take per chromosome. The genome coverage is very low. The whole genome coverage of this marker is very low in case of first generation and second generation marker. That's why we need a marker system should be more automated compared to them, more abundant compared to them, and more genome wide coverage compared to them, more distribution compared to them, and less costlier and less laborious when compared to them. This is in this need there is a development of another marker system called as SNP marker system in after 2000. Why it is 2000? Because at that time Arabidopsis was sequenced, and in 2001 or 2002 at the same time rice was completely sequenced, and by 2010 almost all major cereals full reference genome is available, like become available. Then people started, and in 2010, by 2010, there is a next generation sequencing. Okay, like Illumina came, sequencing become very cheaper. Like per sequence per sample nowadays 250 rupees can able to sequence. You know, within 100 rupees, one species will be get sequenced. Actually, it is announced by China Beijing project. And sequencing become that much cheap nowadays. That's why the sequencing is not a big matter nowadays. That's why the sequencing based markers are coming up. Sequencing based markers becoming tech and boom actually. Sequencing based markers. That's called as SNP. We are going to discuss about SNP. What is SNP? What is SNP means? It's a single nucleotide polymorphism. There are variation among the individuals of a species at a specific positions in the genomes, with each SNP locus defined by Planck's sequence of polymorphic nucleotide. We will discuss what exactly it is. First, I will read. SNP may include either insertions or deletions, and all your polymorphics are not considered as SNP marker. Maybe some mutations also there. Okay, some rare alleles will be there. You are not going to consider. The frequency of this particular insertion deletion mutation or any mutation is above 1% only is considered as SNP. Means, for example, you take in an individual 1 and individual 2, in the place of A, here T will be there, and rest others are similar. Means this variation at single nucleotide level, it is called as SNP. It's very simple to understand. Okay? And this, it is also a mutation. Which mutation? This is transversion or transition. A to T. This is a transversion. All transversion or all transmutation can't become SNP marker. The frequency of this transition or this frequency of this mutation should be the frequency of this mutation should be more than 1% in the population. Then only it regarded as SNP marker. Then each SNP locus can have up to 4 alleles. How it is possible? Can anybody tell how it can be how it could be possible?
Sir, four bases exist. Four bases.
Yeah,
Each base is one
A
allele.
can be changed to T, A can be changed to C, A can be changed to another one ATGC. Okay, there are 4 combination of alleles present per particular loci. Okay, at particular position, 4 different types of mutations are possible. But whenever you say that SNP is biallelic, it's not multi-allelic. SSR is multi-allelic, but SNP we say it is biallelic. Why? Because SNP normally it is tetraallelic. Because 4 allelic variations are possible. But why it is called as biallelic? Anybody knows? You can tell.
At a time, only two alleles it can express.
No, that is not. That may be the reason, but it's not appropriate. Actually, in nature, natural condition or in nature, transition mutations are more frequent or transversion mutations are more frequent. While studying the mutation chapter, in BD Singh, we will read which mutations are more frequent, transition or transversion. This is regularly asked question in ARS. Yes, Abhinay.
Transitions.
Exactly. Transition mutations are more frequent. Can you tell the what is the reason for it?
Because purine and pyrimidine same size.
Yeah, we
Same ring
can.
structure
Yeah,
same.
the chemical
Easily
structure
modified.
is same. If two double ring will changes to single double ring, there will be drastic change in the DNA configuration. Like the structure of the DNA is drastically changed. Maybe it will not going to survive. Maybe there is a drastic changes in the structure or a chemical composition. Maybe that's why the allele may not going to survive many times. That's why purine to purine change or pyrimidine to pyrimidine change are more frequent. Even though 8 combination it can exist, but although only 4 combination transitions are more frequent than the transversion. This is the one reason. Okay, due to drastic change in the chemical structure of the DNA, the particular allele may not going to survive in the during evolutionary nature. That's why purine to purine or pyrimidine to pyrimidine mutations are more frequent. That's why when A more to purine to purine, A to G are normally A to C. Like that, transition mutations are more. That's why you may get only biallelic nature. That is another one reason. Okay. Then the another reason is there will be another reason. Can anybody guess what will be the another reason? Just by thinking you can get. It is also depends on the occurrence of mutation at particular position. It depends on. First answer actually, the first part actually, I think some they told the chances of occurring two existing. For example, this is the one position at fifth base pair changes to T in the second generation. It changes to C in third generation. It changes to G in fourth generation. At same locus, having a mutation from generation to generation. It is highly possible or less possible. At fifth base pair, at particular position only,
Less
occurrence of
possible.
mutation and each generation at the same position, this is a very rare chances. Okay, maybe this may change to this one, but in next generation, you can't say that this will change to this and this will change to this. Maybe we will get this will get reversed. T may be reversed to A may be reversed to T. But T may change C, in next generation C will to change it to G. This possibility is very, very less. This is the another reason. That's why normally instead of SNP, even though 4 allelic combination theoretically exist, but practically it will not exist. That's why SNPs are called as biallelic. I hope everyone clear with this point. Either
Yes,
transition
sir.
or transmission mutation. Transmission mutations are more frequent. Okay. Then the advantages. It's abundant in the gene. Like in Mumbai, actually I was presented a credit seminar on Friday. I was discussing genomic selection in Mumbai. They have taken 292 individuals. 2 lakhs SNP they got. See, 2 lakhs markers. You can't imagine in any marker system. SSR, even though rice is that much advanced crop, yesterday I shown 18,000 markers. The problem in that website, they can able to maintain repository of 18,000 markers. That too in rice. Okay. You can't imagine the SNP and SSR. Here you have 2 lakh marker and compared to in thousands, it is in thousands, it's in lakhs. Which marker is more abundant? Means SNP is more abundant. Means we can get max to max markers. Then more abundance and it is distributed equal. throughout the gene. Means you can't miss any gene position that cannot be mapped. Means it will get high resolution map if you use more number of marker. The density I will explain when we are getting mapping. The density of the marker in the genome is very important. Higher will be the density of the marker and equal distribution throughout the genome is very much necessary for getting good maps. Then frequent in the nature is the SNPs are more in non-coding regions like introns. Okay, and why it is so? Because you know that there is a very less selection pressure, but they can generate phenotypic effects. There is another thing. It's a highly automated and high throughput, and because now the sequencing technologies are completely automated. Either you take Illumina, either you take any other platform, the PacBio, Nanoform, okay, Nanopore, sorry, Nanopore, third generation sequencing. They are highly automated, and nowadays cost point also it's very less. It again depends on genome size, depth you're going to cover. It depends, but as of the small genome crop like Mumbai you take having only 430 MB and rice you take 430 MB. Several having genome size is very less at that time. Sequencing going complete sequencing, whole genome resequencing. It's very approach than the using SSR marker. Then another one of the disadvantage is because nowadays even per base sequence cost is less, but the machine is very costly. For running SSR, you need only thermocycler. It is like in some 5 lakh, 2 lakh you will get. But in SNP marker, one-time investment is very costly. Like you had to bring sequencing machine, it's nearly 1 crore, sometimes 40 lakhs, 50 lakhs, near 2 crores. That's why initial investment is very high. That's why normally what we will do, we will outsource for sequencing. Okay? Then SNP is classified as different classes of SNP were there. Non-coding SNP, exonic SNP, synonymous SNP, diagnostic SNP, genic SNP. But no need to remember anything. Okay, just you remember SNPs. Okay, what is SNP? What is advantages of SNP? And why it is better than surgery? Why it is better than the other marker system? You should know that one. Then you will get. Okay. Then for just your knowledge paper, I am saying non-coding SNP means the SNP is present in the non-coding region. Exonic SNP means SNP present in the exons like cDNA. Okay. Synonymous, these are very important. What is synonymous SNP? Can anyone tell what is synonymous mutation? Synonymous mutation means there is A and in individual 2, there is a change in the nucleotide, but the amino acid you are going to get same. Like it is also like AUG and ATG. But just for example, AUG, even though there is a change in this nucleotide, but both will code for methionine. For example, for sampling means they are synonymous SNPs. Okay. Then there is a diagnostic SNP involved in genetic diseases. Can anyone tell which is the famous diagnostic SNP? Sickle cell anemia. Means there is single transition. It is involved in the sickle cell anemia disease. Like genic SNP is present in the genic region, like exonic region, and anonymous SNP means it will not affect any gene functions. Okay, they are present in the intronic regions. There are n number of types, reference SNP, in silico SNPs. In silico SNP means there are several websites are available. In a database only we can able to mine different sequence SNPs. Okay. It is our in silico based, like you can in database only we can able to identify the SNPs. There is no need to go resequencing. Okay. Then how to identify? I told like there will be SNPs. It is there in the DNA. It can't be visible to our eyes or how. What is the detection system? Means here there will be, you have to go different types. Either you can go whole genome resequencing, genotyping by sequencing. Either there is a arrays, chip based sequencing. There are a number of types are there. Either you can go transcriptome sequencing, exon sequencing. Okay, we'll discuss one by one that one. First, you have to know what is sequencing. Actually, that was the thing. I will explain what is. Actually, this is a sequencing protocol. How we can mine? This is called SNP mining also. For example, you have to go for base calling. Can anybody tell what is base calling? Your reference genome will be there. A T G C A T G C. It is a reference genome. If you are having one progeny 1, then you are sequencing progeny 1, you are aligning all the bases. For example, A T G C A T A G. You are aligning the reads to reference genome. This is called as alignment process. After alignment, you have to go for base calling. For example, A is there, A is there, T is there, T is there, G is there, G is there, C is there, C is there, A is there, A is there. For example, here is C is there. Instead of it, G is come. It's not complementary. Like there will be an SNP. Like this, we will identify the SNPs, aligning your individual DNA sequence to the reference sequence. Okay, this is called as base calling. There are several tools. Okay, BWS software was there, aligner was there, spliceware aligner. There are different softwares are there, but no need to remember. It's just bioinformatics course. Then you have to do quality control. I will explain what is quality control when sequencing comes. Then you have to, oh sorry, here alignment will start. Actually, first base calling means you have to sequence. Then you have to check the quality control, then alignment. Then you have to then again post processing, like remove adapters, remove trim. By using trigonometric software, you have to remove the adapter sequences. Then you have to check the quality score. How to check the quality score means I will discuss that is when the sequencing will comes. Then variant and genotype calling. Then you have to, based on the number of frequency, you will go for filtering of SMP. Actually, I will explain this thing. You should know the basics of sequencing actually. How many of you know what is the base, like before this procedure sequencing? Anybody did this one? No one. Actually, it's a very important thing. Actually, normally first we will use any sequencing platform. Like Illumina, Alexa, like Illumina, PacBio. Then we will sequence in reads actually. We will get read 1, read 2, read 3, read 4 like this. Then you had to assemble these reads. There are two types of assembly: reference based assembly, de novo based assembly. Don't worry, I will discuss in detail. We'll see tomorrow. I think I will discuss reference based assembly, de novo based assembly. What is means? You are assembly the all the reads to the reference genome. After assembly, you will check the quality. Either reads are good or not. Okay, before assembly only, you're checking the reads quality is good or not. Then you go for alignment, like to the reference genome. Like based on the overlapping sequence, you will get an alignment like this to the reference, then it is called as contig. Then after that, you have to check the quality score. Okay, either you're getting sequence like alignment rate is good or not. Quality alignment rate. Like either how many reads are aligned, how many reads are not aligned. After that, you have to go for variant calling. Okay, here A is there, which nucleotide is here. Here G is there, which nucleotide here. Then after variant calling, you will get SNPs. Then you have to check how much frequency. For example, you're having 100 individuals. You're having only one SNP, means 1 by 100, means 0.01. I told you should be more than 1%. Then you remove all these type of variants. You keep only the variants which are having the frequency more than 1%. Then it is called as filtering of SNPs. Then they are called as candidate SNPs. Okay? This is a different procedure, but in nutshell, I told. Tomorrow I will discuss in detail. Don't worry. Okay? Then another thing. What are the techniques we have till today to discover the SNPs? Either you can go with whole genome, specific genome, or a transcriptome. Means you can, if you have more money, you have more reference sequence, then you go for whole genome, and genome size is very less. You can go for whole genome resequencing. But you don't have money to sequence. For example, you take weight. So 18 GB. It's very difficult to sequence whole genome. It's very costly. That's why we are sequencing a specific genome. That is called as genotyping by sequencing. Means reduced representation sequencing. That is called as reduced representation sequencing. Genotyping. It's very less costlier approach than the whole genome resequencing. Either you can use transcriptome sequencing. Only transcriptome means RNA based. Means only expressed part is there. For example, 18 GB wheat genome, only 1 GB is expressed. Most of them are repetitive like this you think. Means cost will be reduced. And what are the SNP are going to getting there are expressed part of the region. That is one advantage from the transcriptome sequencing. Then many times, because this car is very challenging because this is having highly repetitive GNA, and then after you have to do validation. Then how to do validation? It's called as PCR based validation. We have to go for decaps assay, caps assay. I will discuss. I think. Hope I will discuss. Then the emergence of engine technologies made our life easier to discover in SNPs. You can use some EST sequences, express sequence tags. Yesterday we discussed either transcriptome sequencing. Another thing called as amplicon sequencing. Instead of, for example, you having repetitive genome here, you are having repetitive genome. Only amplicons exonic region can be sequenced, and other part will be deleted. That was one approach. There is another term called as SNP mining. Means it's a bioinformatic analysis. This as a protocol discussed. This is SNP mining only. Means you are mining the SNPs from the genomic data by aligning. Okay. How we can do that one? There are like tools like polyfret. There are several tools. Actually, it is there in the book. Just I copy pasted. These are also nothing. Then transcriptomic sequencing means you are using an RNA for the identification of SNPs.
So STS or EST, same thing?
Sequence tagged sites and expressed sites. No, they are different. Expressed tagged sites. Actually, STS may not be expressed. This is from the genomic region.
Yes, sir. You discussed STS, sir, yesterday, not EST.
What?
You discussed STS, sir, not EST.
Yeah, okay. Expressed sequence tags. These are same as like STS only. From cDNA library, you are preparing ESTs, okay, expressive sequence tags. There is a genomic library and cDNA library. From cDNA library clones, we are identifying these expressive sequence tags. Means it's an expressed part because from DNA, where it is, yeah, I had to cover that. In cloning, I had to cover. For example, what is the difference between STS and ES, expressive sequence tag? It is prepared from genomic library. It is from cDNA library. Means here how we can identify the markers means DNA will be there, DNA converted into RNA, then again it converted into cDNA. Here we are identifying the markers. You are tagging the markers here in genome. But in the sequence tag site, we are directly using genomic regions. But many times this marker may fall into repetitive gene. But here not the case. Because in cDNA library, we are not getting any repetitive genomes. No repetitive genomes. Understood what is the difference between STS and ESTs? Means it's the expressed part.
Sir, is that possible? Sir,
Yeah,
is
that
that
is possible.
possible? There is a difference in the polymorphism into introns.
Can you repeat your question? Hello?
There may be possibility that in introns there is a polymorphism.
Many SNPs are abundant in intronic region only. SNPs are more SNPs you are getting from repetitive genome only. In intronic region only, you are getting more SNPs actually. Because exons are mostly conserved regions. Your exons are conserved across the species, across the individuals. But your intronic regions are, they are not more conserved. They are more variable among the species, intronic regions, due to less selection pressure. Due to less selection pressure,
Sir, then if we use cDNA, then there are less chances of polymorphism.
yeah, this they obviously. But what are the polymorphism you are going to get now? They are definitely they are either in the expressed part, maybe they are getting a functional marker. Getting functional marker, it's more probable in case of this ESTs. Okay. I told what is functional marker means the marker is directly associated with the some function of the gene. Getting that type of marker is having higher chance in expressive psychometries. As per the number of markers, you may get less, but what markers you are getting is highly useful. But in STS, you will get abundant marker, but some many are in repetitive genome. Maybe it is less useful than the ESTs. Okay. Now you understood? I think
Yes,
not
sir.
understood. Huh? Yeah.
Yes, sir.
Yeah, these are
Understood.
very tricky. You have to read so many number of times. After reading many number of these many years only, okay, I have reached some level. Okay, some understanding. You need to read so many times. Like it's like a simulation. You have to do trial and error. Then only you will get learn. This is one I was reading on population genetics book today. Was I shared the images? It is from population genetics book only. It's by Hamilton. It's a very good book, Hamilton, Population Genetics by Hamilton. While explaining simulation and models, he just given an example. Like if you want to learn piano, if I teach okay some symbols, you do two fingers, you hold two fingers on upper and one finger on lower side. If I tell any all these rules and regulation, you're going to learn piano or not? By listening to
No,
me,
sir.
no. You have to practice. You have to
Practice.
do trial and error, and you have to practice more time. Then you came up with a mistake, then correction. Then only you will going to learn piano. Actually, then in the same way, actually the population genetics or any genetics concept, it's a like trial and error because all are expectation based. See, nothing is visible. All are expectations and predictions here. The probabilities. Whenever discussing the probability of occurrence of an allele, probability of occurrence of a genotype, they're all expectation. Means they are like after so many trial and errors, then only they came to one assumption, okay, this will be there. Because none of these are visible to our eyes. But they have developed these many concepts based on predictions and exceptions, based on trial and error. In the same way, you have to read like that, practice, okay? Then only you're going to get. Okay. One time listening, it's not like I am not sure that one time listening you're going to get everything. Okay. Then another thing, whole genome sequencing everyone know, but I will discuss. Okay, SNP calling, as I told, the just alignment, checking the variation and checking the frequency of their variation is called as only SNP calling. I will cover what is depth, what is coverage, okay, what is 20x, why it is more than 20x, okay, why you need to go that much depth. We're going to discuss, I think, in tomorrow's class. Then, why we need of all these markers? In first class, I discussed application in plan BD. Why we need all of these markers? So far, we have discussed. See, every marker, what are the things we are going to remember? For example, you should make one table marker, genetic basis of polymorphism, dominant or codominant, locus specific or non-locus specific, and method used to identify the SNPs, and genome coverage. For example, you take SSR. Genetic basis of polymorphism of SSR is either allelic nature. Sorry, allelic nature last. SSR is genetic basis of polymorphism is
Repeats.
number of repeats of SSR. Then it's a codominant. It's a locus specific. Method is normally we use PCR based method and we need sequence information to design SSR. And it is multi-allelic in nature. You take SNPs. Here the genetic basis because nucleotide variation. Because of insertion, deletion, okay? Then it's a codominant in nature. And it's a locus specific. And method is sequence based and biallelic. And high genome coverage and all these high throughput, all those things are common points you should remember. Then you take RFLP. Where is this? Then you take RFLP. Genetic basis of polymorphism, there is a change in the restriction site. For example, in one case, due to insertion or mutation, the restriction site is changed. That's why you're getting different length of fragments. Fragment length polymorphism and insertion or deletion in the restriction site. Due to that's why we are getting different length polymorphism. Then what is the nature? It's a codominant nature. It's a locus specific. Okay, and method is restriction based. It is also multi-allelic in nature sometimes. We'll get more number of restriction sites made. But this nature is not normally not exploited. Then RAPD. What is genetic basis of polymorphism of RAPD? There is a difference in the primer binding sites. Because wherever in some individual it binds somewhere, in some individual it binds somewhere. Okay? That is difference in the primer binding site due to random nature. It's dominant in nature. It is not locus specific. The method is PCR. And the allelic relations cannot be interpreted here. ISSR you take, intersequence. Like this, you should make one table. Then regularly you have to revise it. That is the only way to remember. And all other things are unnecessary. Only this is these are the points are very important. Genetic basis of polymorphism, the nature of the marker, either it is a locus specific or not, and which method is used to identify that marker, and what is the allelic nature, either multi-allelic or biallelic, or you can't interpret the allelic relationship, you should know. Okay, and which is mostly commonly used, and which is most commonly used for high density mapping like SNPs, and which is most regularly and commonly used is SSR, but nowadays SNP is also more regularly used. Nobody is using RFLP, RFID in any lab nowadays. Only few horticultural crops, these markers are still using. Apart from there, other marker systems called SCAR, CAPS, SCAR marker, CAPS marker. Okay, there are DCAPs again. I will discuss that one DCAP marker tomorrow, I think. This is how we have to read the marker systems. Okay, understood what is how to read marker system. Then how to genotype SNP. I discussed how to genotype with SSR marker with my own gel. But SNP, how to genotype with SNPs? How you can sequence each and every individual with that SNP mark? Any guess? There are different types. One is allele specific PCR. Another one is 5' nuclease assay, high density oligonucleotide array chips, bead based techniques, primer extension, invasive cleavage invader technology, multi extend assay, and pyrosequencing. Most of them are outsourced. And no need to read also. Okay, just you remember these are the SNP genotyping techniques. One is very important is CAPS assay. Forget to mention here. Only 5' nuclease assays. Allele specific PCR. This is very important. Okay. You just remember these are the techniques is used to genotype the SNPs, and some techniques are identifying. There are two types: SNP detection, two steps: SNP detection and SNP genotyping. SNP genotyping means these are the techniques. And allele specific PCR is both detection and genotyping. And whole genome resequencing also both detection and genotyping. GBS also both genotyping by sequencing. This is the both detection and SNP genotyping. It can be used for genotyping and detection assist. But these are the techniques you just remember. No need of reading. Parasixing anyhow you are going to discuss tomorrow. But all the techniques you should just know the names. Okay, these are the techniques. If some multiple choice questions came, you should be able to answer. Okay, these are the SNP genotyping techniques. And another, these are two methods. Allele specific, there is a CAPS marker. Competitive allele specific PCR. Okay, there is, you just remember that is one. And DCAPS, derived CAPS markers, also used for the SNP genotyping technique. SNP genotyping of individuals. Okay, these are the things I want to discuss today. What is SNP markers? How we detect it? What is SNP calling? And what is SNP genotyping techniques? Different SNP genotyping techniques. I hope today's class is clear. I think this is the end of only I left with DCAPS and CAPS. Both are SNP genotyping technique. I will discuss after discussing all sequencing platforms. It's associated with sequencing platforms. There are first generation sequencing, next generation sequencing, third generation sequencing. I will discuss tomorrow all this. Okay, any doubts up to here? SNPs, marker systems?
Sir, why SSR are multi-allelic and SNP are biallelic?
The SSR, we can get more number of variations. Okay, that is SNP A, T, G, C. This much only get, and probability of getting these two are very high. That's why it's a biallelic, not tetraallelic. But in SSR, for example, it is 80 repeated 10 times. In another one, 80 repeated 11 times. In 80, another repeated in another individual 13 times. 80 repeated in another 15 times. This is unlimited. But the nucleotide variation is only 4 combination. That's why it's multi-allelic, and that's why you should remember what is the genetic basis of polymorphism. Here the polymorphism is based on the number of repeats. The number of repeats combination is, you can get any number. In one individual it is 10 times repeated, in another individual it is 11 times repeated, in another individual 13 times repeated. These are all different alleles only, no?
Sir,
They
one
having
doubt,
different
sir.
yeah.
Sir, everywhere it is given like S&P shows the maximum polymorphism and it is the best marker. Sir. But
See,
as you said,
maximum
yes,
polymorphism is different, and maximum here the multi-allelic nature of a marker is different. Polymorphism, why? The number of markers are more. Okay, but the combination of alleles only 4 at single nucleotide level. I'm talking. There may be in a single gene how many number of nucleotide is there. If you get how many number of combination of SNPs, that's why polymorphism is more. Overall phenotype is like overall combination of different SNPs are more. When you're talking at individual nucleotide, how many number of possible combination is only 4. That's why it is called as tetraallelic, but the probability of is occurring as 2 is more. Okay, understood. I'm talking at only a single
yes,
nucleotide level.
yes,
In gene, how many number? Maximum, say like your gene having 5 kb. 5 kb means 5000 SNPs, 5000 nucleotides. How much combination of polymorphism you may get? That's why polymorphism is very high. But I'm talking at the different combination is possible. At single nucleotide available, only 4 combination is possible. 2 combination are more frequent, that is biallelic in nature. Actually, this is interchangeably used. Okay, it's not a proper word to use, but it's there in the textbook. We can't do anything. But in SSR, like in one individual, the repeats is 10 times. In another individual, repeat is 11 times. In another individual, repeat is 13 times. Like this, the variation is we can get more number of variation, means we'll get, that's why it is called as multi-allelic nature. Understood? What is the difference? No.
yes, sir.
Another one asking some doubt, Vishwanath, what is the polymorphisms and multi-allelic nature and polymorphism? Yeah, actually, see, polymorphism means, I think the different morphological variation is present. That is only polymorphism. Allelic variations at genic level, gene level you take. More number of alleles for a particular gene, more will be the polymorphism. The red color will go on by one allele, green color go on by another allele, and so many colors go on by so many different alleles at genic level. You don't compare with marker and those things. That polymorphism, that allelic relationships, it's quite different from this combination. Don't intermix, okay? Maybe it is similar, but simply intermixing will make confusions. Okay, any doubts up to here? Actually, I'm ending up.
Sir, dot marker.
Hello?
Sir, diverse carrier technology finished, sir.
Yeah, I will do actually. I had to do that one. After completing the sequencing technology, I will complete diversity array technology. Actually, I had to deal with arrays. Actually, I want to discuss, when I was discussing, you should know what is array-based genotyping, what is chip you have to know. All those things I had to tell. If I tell diversity array technology, you should know what is array, what is chip, how to do sequencing by using chips. All those things are getting confused. That's why I'm not discussing that one marker system.
Okay, sir.
It's easy. If you want to read, read. Okay. It's not, and many times it's not asked in the exam. Okay. Then actually last time the questions were asked very tricky actually last year markers. So very easy, but it feels tricky. Like he asked some if there is a mutation, if there is a mutation other than the restriction site in the genome, that also is very important for the RFLP marker. It's the statement is correct or not. If mutation is not there in the restriction site, it cannot be used as RFLP because if there is a mutation in the restriction site, then only your polymorphism going to get. Based on that, he made several statements like this for each marker system. Then he asked the question, which one is true, which one is false. So you should be able to interpret them very nicely. Okay. Read Akesing and Bidising, which are all taken. I have taken all the sentences, everything from Akesing and Bidising book. Read it. Okay. It's very good book. Practice actually. Without practicing, you're not going to get at a single time. Like at a single time, you can't going to get anything. Read and write, practice, then only you will get. Then any other doubts? Actually, tomorrow I will going to start what is mapping and first mapping populations I will discuss. Sequencing platform I will discuss one day. In one go, genomics I will cover sequencing techniques and advanced techniques, GBS approach. I'll finish in genomics class actually. We'll start with mapping population. Okay. Any doubts regarding this?
No
No
doubt.
doubts means I will close. Okay, okay. Thank you, everyone. Bye. Good night.